I would start to say that this is a work that I've done together with Maurizio, which is my mentor. So he's the only other acknowledgement I will make because it is something we did together. And it is part of my PhD thesis. It is very theoretical, so I'm very sorry. But I try to put as many figures as I can and just a few equations to be very pragmatic. It is you have seen also in the previous talk, the one before me and also the one from Alessandra, that the mean field theories are a central part of neuroscience nowadays for many, many reasons. And then for a theoretical physicist, questions arise, like how can I derive these beautiful theories from first principle, which means from, in our case, from single neurons, and going to the scale that we want, like cortical column dynamics, so neural population dynamics. And you might ask, why would you want to do it by first principle? The answer is because if you manage to do that, then you have a very clear bridge between different scales. So the single neural properties will give you a lot of information about the emergent properties of your system. And this is the reason why. Why we are looking at this. And also, well, let's start, and then I will give you other reasons why this is important. So our starting point, so the first principle, the single neuron is the model of an integrated firing neuron, which I think many of you know better than me. This is a simplified version of what a neuron does, but it is actually quite accurate. And we say that these neurons are integrator. So what happens that we are following the membrane potential of each neurons, and what they do is that they sum all the spikes of their neighbors and friends, and if a given threshold is reached in terms of membrane potential, they will emit a spike, and this spike will be sent towards neighbors, and you will be resetted back to a given value. So those are very complicated systems, actually, even if they are one-dimensional or maybe two-dimensional. And when you put many of them together, very interesting things happen. But what we want is not to talk about single neurons, it's to talk about neural population. So in a way, it makes sense to talk about the distribution of membrane potential inside a given population. And when you look at this and you do the theory, you do the derivation, you find that it is actually possible, thanks to the general concept of mean field, which is, in this case of neuroscience, is quite an extended version of what a theoretical physicist would call a mean field. You can actually derive the time evolution, the equation for the time of evolution of the full distribution inside your cortical columns, distribution of membrane potential. And then in the first movie, you see that the distribution of membrane potential evolves in times. This equation here is very complicated. It is non-linear. And so the only way you can really solve this is via numerical integration. And it is better than doing the spiking simulation. Yeah? No?
May I ask you a question?
Yes, sure.
Why do you have sigma square outside the derivative of the second derivative of nu?
Because
I
we...
don't see very well now. Just think fine.
Because this is an implicit... sorry, so they asked why the sigma square, which I would take the chance to explain what it is the variance of the synaptic current. For conductance-based model, for example, you would need to include it inside the derivative because it will depend on the voltage. But we are considering, this is not a limitation for what I will show you. We are considering current-based neuron, which means that, so you might think that it's the same variable, but it's not. The nu is the firing rate. So I agree with you, they are very similar. But nu is the firing rate of the population. So if you are capable of solving this in some way, you can obtain the firing rate. So the activity of the population, because they are related. So this is what you would want to measure, the firing rate. This is what you have in experiment. But those two things are related in a very complex way. And then, this is a standard question for a theoretical physicist. We don't want the full distribution. It's too much information, and from an experimental point of view, it's very difficult to sample it. So you ask whether it is possible or not to derive low-dimensional dynamics only for the moments of the distribution, which means the average potential inside the population, the variance, or you can be even more creative with that. The firing rate itself, which is something that you want, is one of such macroscopic variables. And so the question is, how can I go from these two different things to a unified theory? To do so in neuroscience, many assumptions are made, and this is not this is normal because the system is nonlinear, so you have to find a way to close your loop in some way. But those same assumptions usually are the reason why the mean field theories works but not so well if you start to look very carefully, because those gives the boundaries of your approach. And what we did with Maurizio through the years, I would say by now, is to derive a theory that does not rely on the standard approximation. And this is the equation that you would obtain. I would not explain what it is, but essentially it is a system for a vector of moments, like the mean, the variance of the membrane potential. And as a byproduct, we derive that equation down there, which is very general, and it is the relation between the firing rate and the moments of the distribution of the membrane potential. This is actually, for us, a very interesting result, because you can very easily imagine experimental setups where you have membrane potential proxy, and you want the firing rate. And there, it's not so easy to find the relation. This is the relation, at least for these models. And or you can have both information, and then having both the firing rate and the mean average potential, you can make a very clear and precise bridge between theory, dynamical mean field theory, and experiment. So this is just to leave. I will leave it there. If you are interested, then we will go back to it. Now, this theory is, now there are all figures, so I have no more equations. And now this theory is very general in the sense that you can decide how precise you want to be, which means how many moments do you want to consider in your description. So what is the dimension of this system? And to show this, I will do something that we very often do in physics, which is the study of the linear response of the system, which can only be done if the input that you give to your system is very weak. But then I will relaxate this in a moment. And so you take the input, you take the output, you make the ratio in the frequency domain, and you obtain something which is called the dynamic response of your system. And it is a function of the frequency of the input, right, in the Fourier domain. And you see in different colors, starting from red to black, what happens if you increase the dimensionality of the theory. Black is the full population dynamics, so the infinite number of moments. And you see that already with 2 moments, a 2-dimensional system, you can capture very well the low frequency regime, the resonance peak, which is around the stationary firing rate, for those who care, and the high frequency limit is not that bad. And you can get better by concluding 3, 4, or 5 moments. But this is like cheating, because this is linear response. It's actually quite easy from a theoretical point of view to take a very good linear response. The problem is that neural populations, as you've seen before me, works outside of equilibrium. The real concept of reversibility that you heard before is because those systems are out of equilibrium. They are non-linear, extremely non-linear, because you wouldn't have limit cycle and slow wave if they were linear in the shape that you see. And so linear response doesn't really give you too much. And this is what you would obtain if you put yourself in an extremely nonlinear case in which your population is capable to produce a limit cycle, so it is a kind of a slow wave in a sense by itself. But you also add an external drive which is very strong. And you cannot really see with this resolution, but you have in red the result for a 2-dimensional moment system. Then you can increase by 1 with the purple, and the black is the full Fokker-Planck equation. So they are very similar, I would say. But from this, you cannot really see what's the difference. So this is just to show that we have this kind of control with this theory. Now, in the last part, I will try to give you something about this nonlinear response, which actually we found with Maurizio, it is not very often studied in neuroscience because it makes sense. You cannot do analytical calculation from a theoretical point of view, but still you should study it. And to do so, let's take this concept from dynamical system theory. This is what we would call the Poincaré section of your data, which is very simple, actually. You have your dynamical system, your population of neurons, which has a complex trajectory, and instead to look at the whole trajectory in the whole dimension, you take and look at a very specific region of points that you define with a given criteria. And you look only at the evolution in time of that specific point. This is very rigorous, by the way. You can prove that the two things are extremely correlated. So it is a kind of reduction of information. But in the context that we are looking at this, this is what I define as a Poincaré section. We have that black finite rate. Sorry, I will try to. This is your finite rate in this very complex scenario. And we will only consider for a moment the local maxima that you see here. So the distribution of local maxima as a function of the external input. Now, if you study dynamical system, you have seen something like this before, but this is the case that you can actually do in experiment in which you modulate your cortical column with an external field. And if you do so, you are basically you are changing in time the mean of the synaptic current. And what you can see here is this Poincaré section of before. I will explain what you have. On the x-axis, you have the external frequency. And on the y-axis, you have the distribution of points of local maxima, which means that when they are very simple, like 1 point, it's a very simple trace. When there is a crowded region, this is a region in which your system is aperiodic. It might be even chaotic, is irregular, because you have many, many maxima. I will show you some example after this. But the point is, once again, that this is the full population dynamics response, nonlinear response, out of equilibrium response, and this is obtained by just 2 moments, the two-dimensional system of before, the theory of before. And then we can be fancy, and let's say that we found a way to modulate not the mean of the synaptic current, but the variance of the synaptic current, some way. And you find a completely different nonlinear response, a very crowded, many crowded region with some very specific intervals. Those are not by accident. They are very specific frequency. But once again, the two-dimensional system captured qualitatively very well the full nonlinear response. And then this is just to show that if you modulate the mean and the variance of this very particular system, you obtain something completely different, which is something that you cannot really tell if you look at the theory of linear response. For example, if you look at the work by Brunel and all this framework. But this is just maybe for the theoretician, so I will leave it there. I will conclude by showing, of course, I do not have time to really go into this many things that we found. We think that all of these things are new, and so there is also a lot of physics involved. But I pick just one to show you something interesting, which is the following. Let's say that we fix the frequency and we start to change the strength of the amplitude. We can do this because our model works very well even when you have a very strong input. And this is what you observe if you increase the strength, right? It changed completely. And you start to have a very complex patterns. So also the strength of the simulation can tell you a lot about your system. This is a neural network. And you are forcing it with a very strong stimuli. Now let's focus at this specific frequency. I'm sorry, it's very hard to see. I will tell you what you have at that specific point. You start by a single point in the Poincaré section, so a very simple trace. Then you increase the strength, and many, many peaks appear, so you increase the complexity of the trace, and you increase even further the strength of the stimulus, and they disappear, a single point. And this is what you see if you look at the finite rate. Once again, this is the low-dimensional system, and this is the full population dynamics. So you start with a given strength of the stimulation, fixed frequency, it's very simple. Then you develop this kind of envelope, and you start to see many different peaks, and then there is an abrupt change, and disappear completely. This is something that you know very well. We talked about it. It is a bistability of the system, but it's a very weird one, because you typically look at bistable systems that are two different fixed points. So the finding it will be, yes, fluctuating, but around a given value. This is actually something different. It means that you have two different stable cycles of different shape. And to show this, we did an hysteresis protocol. This is the characteristic way you want to highlight this effect. So we start with a given value of strength of stimulation, and we have this very complex pattern. Then we increase the strength, and then after a while, we go back to the original point, and you have something completely different. Much simpler, but the point is that there are two different limit cycles living inside of this. This is actually the story. Of course, I had to be very concise, because I don't have much time to explain all the interesting things that we found, but it was more about, the focus here was more about how this low-dimensional theory, which is very general, it can be applied to current-based, to conductance-based. For example, for the leaky integrated neuron, is completely analytical, so it is also convenient for a theoretical point of view. And it can explain very well out of equilibrium dynamics, strongly nonlinear, open system, which is the most complex case that you can imagine. And this is typically the time when the low-dimensional description of mean field theories breaks down for the assumption that you have to make to reduce the system. In this case, they do not. But we found in the process many interesting new phenomena that have to be studied. It's an open world now, like hysteresis of limit cycle and so on. And I conclude now, I finished, just saying that this has many implications, I think. One is, of course, about the physics and then the neurobiology of this, the behavior of this system. There is a lot to be said that it hasn't been said before. So it is a nice tool to do that. And if you think about full-scale brain simulation, like the one you see before, where each single node is one object like this, you start to think that maybe it's very important to understand the limits of the low-dimensional description that you put in the single nodes, because they build up your full-scale simulation. And that's it. Thank you very much for the time. And thanks, Maurizio, for everything he has done and the help that he gives to me for this work. Thank you.